Hi, my name is Selim Kalayci, and I will be presenting our study titled ProNetView-ccRCC, a web-based portal to interactively explore clear cell renal cell carcinoma proteogenomics networks. Other researchers involved in this study are Francesca Petralia, P1, and Zeynep Gümüş, and we are all located at Department of Genetics and Genomic Sciences and Icahn Institute for Data Science and Genomic Technology at Icahn School of Medicine at Mount Sinai. Here you see the overall poster for our study. Now I would like to zoom in each section and give some details for you. First, I would like to provide motivation and background for the study. The National Cancer Institute's Clinical Proteomics Tumor Analysis Consortium, CPTEC, has been performing comprehensive proteogenomic characterizations of several types of cancer. And to identify drivers of cancer progression from these massive datasets, co-expression patterns between protein pairs are being characterized, and these are easily represented as networks. Furthermore, these networks increasingly borrow information from other types of datasets generated on the same samples, which result in large and complex networks that are challenging to communicate visually. To quickly and easily explore and interpret these networks and associated metadata, we need effective visual exploration tools. For this purpose, we developed a user-friendly portal called ProNetView-ccRCC. Using ProNetView-ccRCC, you can explore two types of networks. First one is the phosphopeptide co-expression network of ccRCC tumors, and second is the 30 network modules derived based on topology of the co-expression network. Now I'd like to list some of the main advantages, capabilities, and technological features of ProNetView-ccRCC. ProNetView-ccRCC is custom built based on CPTEC committee needs, and it incorporates multiple CPTEC-specific analysis data. It's also easy to access and use. It runs on any standard modern web browser without installing any additional plugins or software. It utilizes 3D exploration capabilities, which is critical for dense and complex networks. So using ProNetView-ccRCC, you can explore and interact with CPTEC networks in 3D. You can view clinical variables associated with each gene, filter genes across entire network, view list of enriched pathways for network modules, and display the genes associated with each pathway. And you can export network visuals as PNG images and download network inputs and clinical metadata information. For the development of ProNetView-ccRCC, we utilize standard HTML, CSS, and client-side JavaScript libraries. We also utilize 3.js framework for interactive network visualization. We also adapt and reuse algorithms available in IK. Here you see a screenshot from the landing page of ProNetView-ccRCC. You see a 3D network view. You can rotate and zoom in and out of this network. On the right hand side you see the modules listed, and below that you see a search box. Here you can search for a gene of interest such as ERG. Once you search for that gene, it is highlighted in the network as in red color. Also associated metadata are displayed, as you can see, such as the connecting genes and the association with clinical variables. You can also notice that ERG is located in module 26 angiogenesis enriched module. To investigate further, you can click on the name of this module. When you do that, you can get a visual such as this. In this module network, you can explore the network in more detail. In this network, the node size is proportional to the number of connections, so you can identify the hubs more easily. Also, clicking on a node displays the name of the node along with connecting nodes and associated clinical variables. In addition, you can perform other investigations in module networks. On the left-hand side, you see the phenotype search toolbar. You can specify a phenotype of interest, and you can specify an FTR cutoff. After you submit, you will get the list of genes that satisfy this criteria in the text box, as well as they are highlighted in the network in red color. And on the right-hand side, you see a table listing the enriched pathways associated with the module network. You can click on a pathway name, and the genes associated with this pathway are listed in the text box. as well as they are highlighted in the network in the red color. ProNetView-ccRCC is an ongoing effort. Currently, we are in the process of incorporating other ISCB networks across different cancer types, data types, and tissue types. Similarly, we will incorporate clinical analysis data associated with these networks. We will also provide additional functionalities to improve network exploration capabilities, including network topology-based clustering algorithms, edge bundling algorithms, support for hierarchical network exploration, and metadata query capability across all datasets. And I would like to acknowledge the grant support from NIH, NCI Clinical Proteomic Tumor Analysis Consortium SPTAC. I would like to also let you know that we love feedback and we welcome any kind of feedback such as questions, issues, additional feature requests, and please let us know. You can see our email addresses here. You can reach us, and this is the URL. Please go ahead and explore the tool and let us know. Thank you.